Hello everyone. What we're going to talk about in this third biochemistry lecture is some basic biotechnology techniques that are used in the analysis manipulation, particularly of the deoxyribonucleic acid, so DNA. So this is what we're going to talk about in this lecture. First, we'll talk about this enzyme. They're used to cut DNA, which are called restriction enzymes. Then we'll move on to an example of how using this enzyme allows you to build some sort of map referred to as restriction maps of DNA fragments or DNA molecules. Then we'll talk about how DNA molecules can be separated by electrophoresis and how they can be visualized. Then we'll talk about how DNA molecule can be combined, forming what we call recombinant DNA molecules in order to do different things, to increase the number of desired DNA molecule or to express a gene. And then we'll finish up talking about this very common technique found in several labs, which is polymerase chain reaction, which can be as is or in certain modifications like RT-PCR. So let's start talking about restriction enzymes. Before we talk about restriction enzymes, all enzymes that act on nucleic acids, so both DNA and RNA in general, although they will be specific, they're all referred to as nucleases. Okay, so any enzyme that hydrolyze, and basically what they do, they cut the phosphodiester bonds. Okay, now depending on how they act, physically speaking, they can be divided into endo and exonucleases. So an endonuclease will cleave the DNA from within. Endo means inside, so it will be chopping basically the DNA from the inside, not from the external side. Instead, some enzymes, like if you remember, DNA polymerase 1 has exonuclease activity, will start degrading the DNA strand, the RNA strand, the primer, from the end. And they would do that exactly by starting from the end of the primer. So any enzymes in general that acts from the end of a nucleic acid will be referred to as an exonuclease. So when we talk about restriction enzyme, we're talking about endonucleases. So they are one subset of endonucleases, which are commonly used to do different things, to analyze DNA sequences, to recombine DNA. So you can cut out things with this enzyme and then stick them together within another molecule and so on. So these enzymes, they are actually natural enzymes. They're normally produced by bacteria, and they have some sort of immune system function in bacteria. So what they do, and that's where the name restriction endonuclease comes from, they do restrict the entrance of these bacteria-specific viruses, which are bacteriophages. So what they do, once they attach to the bacterial cell wall and cell membrane, they would inject their own DNA. So bacteria do produce different types of restriction enzymes that actually do cut, work as endonucleases, and they cut specifically the bacteriophage DNA. Now, bacteria have their own DNA as well, right? They have their own chromosomal DNA, but they can protect themselves from their own restriction enzymes through methylation. If you remember, we talked about that when we were talking about nucleic acid biosynthesis. So their own DNA is methylated, and so the restriction enzyme cannot act on the bacterial chromosome, but can still act on any foreign DNA molecule injected into the bacteria, such as the bacteriophage. Now, what makes them unique is that these enzymes, so there are many types, okay, but they all bind, first of all, to a very specific sequence in the DNA. So they recognize only a specific sequence which can be 4 to 8 base pair longs. This one is 6 base pair long. Not only that, but their sequence is always a palindromic sequence. What do we mean by a palindromic sequence? We mean that it's a sequence that can be read in the same direction. If you read the two strands in the 5' to 3' direction, like this one right here, you see is the sequence is the same. GAATTC, GAATTC. Same thing. So if you read this strand 5' to 3', this one 5' to 3', you will get the same sequence. And all of them are of the same type. If you want to test yourself, just read any of these sequence, we'll see that they are palindromic. It's similar to many words, like the one, and a best example I could come out with is my hometown basketball team, Reyer. It's actually the current Italian champion for female basketball in Italy. It was the male champion last year as well. So it's big team in Italy. But if you look at the name, it's a palindromic name, exactly the same thing. Now, if you read this name in one direction, Reyer, or the other way around, this is actually a last name of the person that found the team, this name will be referred to as palindromic. So, but when it comes to nucleic acid now, it refers to the sequence of the two strands in that specific restriction site, that if you read them 5 to 3 prime in one strand or the other will always come out the same. So this is a palindromic sequence. You can see here, same direction, same sequence of nucleotides, same this one, but this one might be look deceiving. It looks almost like, but it's not. In fact, is not a palindromic sequence. It doesn't read the same way. This start with a G, this start with a C, right? And many other things are flipped around. Now, so restriction enzyme bind to these palindromic sequences, which are 4 to 8 base pairs long. They're very specific for those sequences, and when they bind them, they cut the DNA at that sequence. And what they do, they cut the phosphodiester bond in both strands. So one phosphodiester bond for each nucleic acid strands, for each DNA strand. The way they cut it leads to the formation of two big types of ends. Some restriction enzymes, like this one right here, HAE3, it cuts the phosphodiester bonds in this sequence in its own restriction site here, which is again palindromic, only 4 nucleotides. It cuts the two phosphodiester bonds at the same level. So when it does that, now it generates obviously an opening in the DNA that doesn't have any partial single strand DNA molecule. So these are referred to as a blunt end. Other restriction enzymes like EcoR1 or HIM3, very common one to be used in the lab, instead they cut the phosphodiester bonds where they cut in the two strands, they're not aligned with each other. So they're actually offset. Like in this case, there is 4 nucleotides in between. And so once they do that, now the DNA, the two fragments of DNA that are generated, now they would have some partial DNA, single strand DNA sequence, which are still complementary to each other. They can still stick with each other. Hence, these are called sticky ends, because the two end sequences produced can still stick to each other, and again, they have a partial single strand DNA molecule there. Now, what is the function? What do you use restriction enzyme for? They can be used for many things, right? They can be used to make DNA molecules linear. You'll see when we talk about gel electrophoresis later on, how molecules of DNA that are circular, they can have different shapes. It's the same molecule, even the same size, right, can actually acquire different shapes. And so you can use the restriction enzyme to do one cut, and instead of keeping it circular, you can make it linear. You'll see the reason why you would do it in an agarose gel later on. Other thing you can do, you can analyze molecules. I'll go through an example in a few slides of a restriction map. How can you generate a map of a sequence of a fragment of DNA by figuring out where different enzymes can cut, and has an advantage I'll explain in a second. And then you can produce recombinant DNA. So you can use similar enzyme to cut one fragment of one molecule, opening up another molecule, different molecule, and then put them two together, forming recombinant DNA. We'll talk about that when we talk about DNA cloning later on in the lecture. So first example is the use of restriction enzyme to do what we say restriction mapping. Well, the point of restriction mapping nowadays is less and less frequent to do this. But in the old days, it would happen that, let's say you're studying a gene or a promoter, and you're trying to figure out exactly where in the genome is, and so you would take some part of the genome that you know contains your gene or promoter, for example. You would cut it down, and then you would end up having some sort of fragments. Now you start having evidence that, for example, your promoter is within these 7 kilobyte fragments. it's important to know in those days, in the early days, we're talking about 20 years ago, it would be very expensive to sequence. 25 years ago, so it would be very expensive to sequence the DNA. So you had to rely on another way to try to come out with some sort of mapping there instead of having the sequence. And so you could do things, use the enzyme. So you would try to figure out, okay, which enzyme can cut in different part of this molecule. When you do that, first of all, you get some information about the sequence there. There will be these palindromic sequences, but also you can figure out how you can chop, ultimately, by knowing exactly where this enzyme will cut, how can you chop this fragment smaller and smaller so you can narrow down even more to find out where, for example, in this case, where your promoter is. So how do you do the restriction mapping? What you would do, you would take a fragment like this one, 7,000 base pairs, so 7 kilobases, and you will cut it with different enzyme, as is been done in this example here. And then you would take the product of digestion. So in a test tube, you would take some of this DNA, you would cut it, say, with HIM3. In another test tube, separate one, you will cut it with this restriction enzyme, SAL1. In another one, a third test tube, then you will combine these two restriction enzyme. And then you would take the product of digestion of each test tube and you will run it through an agarose gel. Now, just anticipating something that we'll discuss later on, DNA molecule, once they are linear, they can run through a gel proportionally, and the rate of migration, so how fast they go, it's proportional, inversely proportional to their size. The smaller, the faster they go. Okay, so they can be separated based on their size, which can be ultimately determined by comparing it through a standard curve generated using a mixture of DNA molecules that have a known size. So that's why we refer to as a marker. This will be a mixture of molecules that you know one of them is 10 kilobases, the other one is 8 kilobases, and so on. So now, going back to restriction mapping. So you will run the product of digestion, and then you will use this information to try to figure out where HIM3 and SAL1 are found within this fragment of DNA. So how would you do that? Again, you would analyze the products that you have there. So now, you know that the total fragment here, once it's not cut, it's a linear fragment, is 7 kilobases. That's what we know. Now, where does HIM3 and SAL1 cut in this? So can we place these two restriction sites in the restriction map and generate a restriction map here? Well, now, first you look at one of digestion at a time. First thing, HIM3. Now, the gel shows us that this 7,000 base pair band, once is digested with HIM3, breaks down into two fragments, a 6.2 kilobases, so 6,200 base pairs one, and a 0.8 kilobases. Obviously, the sum of the two will give you the total length of this fragment. Well, what this is telling us is that HIM3 must be cutting 800 base pairs from either end, either from this hand, as you can see in this example, so we'll be cutting right here. This will explain what you see here. Or it's possible that it could be cutting from the other end, and you will still get the same result there. You can't really differentiate right now in this. So both of them could be accurate. Just for simplicity, I'm gonna focus now for the reasoning on this map. But both of them will be reasonable. They would just give opposite results in the final version. So let's focus just on this result. Now, let's analyze what happens when you treat the same fragment with this restriction enzyme, SAL1. Now, SAL1 generates a 5.8 kilobase band and a 1.2 kilobase band here. So the total gives us still 7 kilobases, meaning it's cutting only once again, like in 3. And now we know that it's cutting a 1.2 kilobases from one of the end. So either this hand or this end right here, right? How do we know where SAL1 cuts? Does it cut here or does it cut here? Well, we can figure it out by looking now at the result of the digestion with the combination of both restriction enzymes. So if you look now, what you see here is that of these bands that were generated by SAL1 alone, once you add on top of that HIM3, the 5.8 kilobases band doesn't really disappear, it stays the same there. What it means is that HIM3 for sure is not cutting within the 5.8 kilobase band, right? But what you see is that now the 1.2 kilobases fragment here is the one that gets digested by the him3 into a 0.8 and a zero point 0.4 kilobases, which obviously when you add together will give you 1.2. So telling you that this him3 is cutting within the 1.2 kilobases, so pointing to us that then Sal1 and him3 should be close to each other at a 0.4 bases distance. Now, when you combine together with these results, and again, could have been possible the opposite. So if him3, you place him3 here, then at this point, Sal1 will be on this side as well. But when you combine it together, now you can come out with a restriction map where you indicate where the two restriction sites are located within that fragment. So what you would do in a restriction map, you will label as 0 the starting site. So in this case, we label this one as 0. Now we label at which distance each of the enzyme will be cutting. So him3 will be cutting at 0.8 kilobases, so 800 base pair from the beginning, if this is 0. Sal1, 1.2 kilobases from the beginning. So the distance here, I know if I digest this fragment, I want to get just this part here, I have to combine the two, and I would expect to get a 400 base pair fragment here. And then from Sal1 to the end, there will be a 5.8 base pair fragment generated there. What is the point of doing this? Again, you would do this not only with the enzyme, but we would try to cut all of these different with many other enzymes here. So once, if you had a very accurate restriction map of this fragment, now at this point you can start chopping this fragment, exactly knowing what to expect, clone the different parts, and see where within this large sequence there is actually the one where the promoter is in our example, or if you're looking for a gene, where the coding sequence of that gene could be. All right. So by having a restriction map, it would allow you to focus more and more on shorter sequences instead of large ones. Now, we'll go through some other example of restriction mapping on plasmid circular molecule DNA during one of our live meetings. So I'll stop here for restriction maps. What we're going to move on now in this next part is how do you separate and how do you detect DNA molecule. So we talked about restriction enzyme, how they can cut the DNA, and how we can use it for restriction map. And just for example, when we're talking about restriction map, we talked about agarose gel, right? Obviously, you don't chop DNA off just for the sake of it. You want to also separate the fragments. So how do you do that? Well, many molecules, not only DNA molecules, can be separated using what we call electrophoresis. Electrophoresis is a technique, or a scissor technique if you want, that allow to separate charged molecules, okay, molecules that either have a charge on their own or they're given a charge by adding another molecule, but separate charged molecule by putting them into electric field. Okay. Now this could be DNA, you know, that have phosphate group, they're negatively charged, and so they can be separated and they will move towards a positively charged pole. Proteins can have different charge depending on the residues that they have and the pH of the solution. But this molecule could be separated with this technique, right? And now these techniques can be used for different purposes. You can just use it to see if something is present. For example, for proteins, you want to know if a tissue, liver, expresses a certain protein that is found in the brain, or is only the brain doing it. So you would extract the protein from the liver, you will run them on a gel, and then in that case you could use a specific antibody to visualize that specific protein to see if it's expressed or not. That would be called a Western blot. Could do a similar thing with DNA, we will refer to a Southern blot. Or, for example, you can see if there is a sequence of DNA in the genome. You amplify by PCR, polymerase chain reaction. We'll talk about that at the end. And then you run it on a gel and you see if that gives you anything or not. Is that sequence present there? Another reason why you want to run a gel electrophoresis is to determine the size of a molecule. That's exactly what the example here. By running here this molecule together with a marker, again, a combination of molecular DNA with known size, you can actually figure out the size of these molecules. The third purpose of running an agarose gel, again, here, is to separate these bands. You're separating these bands. You could be interested in actually on separating this band and cut this one out if you're doing cloning. So you can even separate them physically and then purify one molecule. So there are different purposes of doing different gels here. Now, there are different type of gel, okay, or electrophoresis can be done with different type of gels. Now, the two main ones that are used are the agarose gel, which are made with agarose, and then there is another one which is called polyacrylamide gels. Now, the difference between them is on the size of the pore they create. So both of them, the function of this gel in electrophoresis ultimately is to slow down the migration of the charged molecules once they're put in the electric field. So imagine DNA molecules, again, are negatively charged, right? So if you put them in a buffer in between two poles, a negative and a positive poles, so you create an electric field, those molecules will go towards the positive pole. If they are in a buffer, they will go super quick, in a fraction of a second. They will migrate. So you can't really separate this molecule at that point. So the molecule, despite their size, shape, and whatever, they will move all at the same rate, super fast. But you don't want that. You want to be able to separate them. And so to do that, that's exactly why you add these gels. And these gels, they form these networks, right? Either of agarose or polyacrylamide, through which now the molecule have to move. So imagine you are a molecule of DNA, you have to go through these holes. This creates resistance and slows you down in your migration towards the opposite pole. Okay. Now, the difference between agarose and polyacrylamide gels is on the size, ultimately, of this pore. Polyacrylamide forms smaller pores than agarose gels, and therefore allows to separate smaller molecules. So if you're talking about a 10 nucleotide molecule, 20, even 80, you would use a polyacrylamide gel. If you're going higher than 100 to 200, then you can use the agarose gel. You can separate something which is 1,000, 10,000 base pairs with an agarose gel. The difference that you can even visualize with a polyacrylamide gels is up to 1 nucleotide, and in fact these were used for DNA sequencing to see the specific technique. So they're extremely accurate for very small type of molecules. Now, what affects the migration during a gel? And this is important for what you'll be doing in the lab. You'll be running a gel, an agarose gel in the lab. In theory, there are 3 factors that would affect the migration of any charged molecule put in an electric field through a gel. These are the factors. One is size. The smaller molecules compared to a larger one would move more easily, obviously, through the pores compared to a large one, right? In this case, this may be a bit too large for the example, but just to give an idea. Obviously, this one, the smaller molecule, the faster it can go through these pores, right? So, makes sense. Size is a big factor. Second thing is shape. Now, look at this molecule here, group with the green line here. They actually have the same size. They are exactly the same size, but they have different shapes. Which one do you think will move faster? Well, I believe this one. This one, overall, it's more streamlined if you want to go through these pores compared to this one. So the shape is the second factor that affects that. And obviously, the amount of charge that you have per unit of mass. So if 2 molecules, like this one, they have exactly the same shape, same size, but this one has more negative charge, and here you have a positive pole. Obviously, these will be attracted to this positive pole much faster than this one. And that's what we mean by the ratio between charge to mass. In this case, the mass is the same, but this one has much more charge. So the charge to mass ratio is in favor of this one, getting it to move faster. So these are the 3 factors: size, shape, and charge to mass. When it comes to DNA, and that's what we focus on here, in reality, charge to mass ratio is not an issue. Why? Because if you think, what is carrying the charge in nucleic acid in general, and in this case on DNA, is the charge carried by the phosphate group. So each nucleotide, right, will be carrying two negative charges. This one too usually is negatively charged. So we'll be carrying the charge here, right? And so you would have two charges, sorry, one charge here for each base pair. So two charges, sorry, for each base pair. So if a molecule like this one, right, has 4 base pairs, you would have the mass, assuming that is 8 in base pair, you would have 8 over 4, right? The charge per mass will be 2. Now, if it had only 2 nucleotides, now the ratio will be the same, because at this point you would have 4 negative charges. For 2 nucleotides, the ratio is still 2. So the charge to mass ratio now for any DNA molecule will be exactly the same. Okay, identical. So it's not an issue in migration. So if you have a 100 base pair DNA molecule or a 501, that ratio will be the same. So it will not be affecting the rate of this molecule moving through the gel. However, what is still affecting the DNA is the size and the shape of the molecules. Here you have an example that shows you how the shape affects the migration of DNA. Now, this molecule right here is what we call a plasmid. We'll talk about that later on in this lecture. These are extra chromosomal molecules, circular molecule of DNA that you find in bacteria. They're circular. But now, if you extract this plasmid from a bacterium, take them out, now this molecule could have different level of tertiary structure. You remember when we're talking about the structure of nucleic acid, we're talking about how the nucleic acid, they have a primary structure, which is the sequence of nucleotides, secondary structure, which is the alpha helix, and then they might be supercoiled. That's exactly the example we're talking about here. Now, the mixture of plasmid from the same bacteria that you have extracted from the same bacteria would exist with different level of supercoiling. This one level is getting more and more and more supercoiled. So now you have that the same plasmid in terms of size. This is exactly, if this is 3,000 base pair, it's 3,000 base pairs here, 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 here. But now there is a different shape there. And you can see it. If you take this DNA as is and you run into a gel, agarose gel, you will see that now there's different forms. So the circular one here, non-supercoiled, will run a certain level. If it has been cut, it will run a certain level because you have changed the shape at this point from circular to linear. And then if it's supercoiled here, will run even faster. Now, how can you overcome that and make sure that all of these molecules can move at the same rate? Is by getting rid of the difference in shape. And how can you do that? Exactly with restriction enzymes. If you take this molecule and you cut them all with the same restriction enzyme, let's assume EcoR1, one of those that we mentioned before. Let's assume it cuts only once. You cut it here. It will cut these too. They are all the same sequence of DNA. All of them at this point will be made linear. The supercoiling will be lost, and all of them would have the same shape, and therefore they will migrate all at the same level. So, going back to the 3 factors there, we have that affect migration in an agarose gel, and focusing on DNA here. So we have the size, in this case will be size in base pairs, the shape, but if you cut the DNA with a restriction enzyme, you lose that factor, and the charge to mass ratio, which is not an issue when it comes to nucleic acid, including DNA. So if you digest, and that's what you'll be doing in your lab, you'll be digesting the plasmid DNA that you will isolate, and the reason why you digest it is exactly to make it linear. So its migration at that point would be not dependent on the supercoiling anymore, and therefore the shape, but will be just proportionate to the overall size of the DNA molecule. All right, and you'll do that by running a gel. In your case, will be an agarose gel, would have a specific concentration, and the concentration is used depending on the size that you're expecting. If you're expecting very small molecules, usually you go with a higher concentration, even 2% of agarose. If you expect big molecules, then you can go with a lower one, 1.2% of agarose. We'll discuss this in the lab. Then, though, once you have digested with restriction enzymes, so your plasmid, and you want to separate your molecules, then you have to visualize them. In general, beside restriction enzyme, DNA can be visualized on a gel in different ways or outside of the gel in different ways. The two main ways are by either using a radioisotope like P32, radioactive P32, very high energy gamma radiation there, or by using fluorescent, much less dangerous. So when it comes to radioisotope labeling, basically your DNA before being loaded onto the gel would have been labeled by using some radioisotope containing deoxyribonucleotide. So synthesizing some of these nucleotides would have been inserted into the DNA, and now you would have radioactive DNA. Then your DNA will be loaded onto the gel, ran. It wouldn't be visible by the naked eyes. In fact, you would actually be wearing also goggles and everything, super protective gears in this case with P32. But then you can visualize where the DNA molecules are by taking the gel and doing what is called an autoradiography. So you take a film, you leave it there for 30 minutes, an hour, depends how much radioactivity you expect to have there. And then wherever there is DNA, there will be radioactivity, and this will be forming a black dot or a black line on the film. You develop the film, you see exactly where the DNA has arrived to in the gel. So that's the first way in which you can visualize the DNA. You can make it radioactive with P32, S35 is another way. For example, for proteins, you could do that. And then you run it on the gel, and then you visualize it with a film. Second way is instead is done after you separate the DNA. So in this case, the DNA doesn't have to be treated before you load it, and that's the way you'll do it in the lab. You can load your DNA, the product, for example, of digestion, in a gel. You can add either directly to the gel or after to the buffer. You can immerse the gel into a buffer that contain a fluorescent molecule that binds to the DNA. These are usually intercalating agents, so molecules that do bind in between the double helix. Okay. Now there are different types. The historic one is ethidium bromide. It's a red-looking compound. Well, then there is red safe too. This is considered mutagenic, so now it's being replaced more and more with red safe. In your lab, you should be using red safe, which is not considered mutagenic, but I will still use anything that binds to DNA as a potential mutagenic compound. And so you can add it either to the gel or to the buffer in that. And so after your DNA has ran, now this chemical would tend to bind to the DNA and therefore accumulate where there is DNA. How can you make it visible? Well, it's based on the property of this compound. This compound can actually, if you irradiate them with UV light, they can absorb UV light and then emit visible light. And so if you take this gel, now you put it under UV, now the UV light will be hitting the gel. Wherever there is accumulation of the ethidium bromide or red safe, you will start seeing a visible band, and therefore you will know where the DNA bands are. And then you can go on and quantify their size and so on. Now, when you run DNA that's been digested, so made linear, as we say before, is running proportionally to the overall size. And therefore you can use gel, as you'll be doing in the lab, to determine the size of any DNA molecule that you load in the same gel. And the way you do it is by running in the same gel a mixture of DNA molecule of known size. Now, the migration of the molecules of known size here will be proportional to the logarithm of their mass. So if you take the logarithm of 100 base pair, 200, 300 here, and then you measure the distance, how far these bands have migrated from where you loaded them, then you can generate a standard curve, so which correlates again the relationship between how far these bands have migrated compared to the logarithm of their size. And then you can generate a standard curve that can be used to quantify the size of any molecule present in the same gel. For example, this molecule right here all you would have to do is measure the distance of migration of this molecule, enter it here, see where it meets the standard curve here, infer the logarithm, and then you could infer the logarithm of the size of this molecule. You do the antilog, you determine the size. Okay, you'll discuss this in more detail in the lab, but that's another application. So the gel can be used to separate molecules, to measure them, like in this case. It could even be used to separate. Imagine this being a mixture of a product of digestion. You could actually cut out this band and therefore have separated from the other mixture there. Okay, so we talked about restriction enzyme, how they can cut DNA. We talked about how you can use them to do restriction mapping, and then we discussed how you can separate the product of, for example, in this case, of the digestion restriction enzymes or any molecule is charged. What is the point of all of this? The point of all of these is to manipulate DNA, okay, to produce molecules, which are what we call recombinant DNA. Before we get there, let's talk about cloning. What does cloning mean? Cloning is a generic term, okay, and when it's applied to living organism or in biology in general, refers to making millions, billions of identical copy of one specific element of a population. For example, you take a virus, you put it with some cells, you get it to grow within this cell, so it reproduce. That's cloning a virus. You are making millions of identical copies of that virus. You can do that with cells. You can start with one cell, allow it to grow, and you make millions of copies of that, like bacteria. We do it all the time. You can do it with organism, Dolly, right? This is an identical copy of the mother. It was taken, the nucleus was taken from a somatic cell from the mother, put into a enucleated egg, and then you got this beautiful sheep. So that's another example. And then you can do that with DNA. The same thing applies to DNA. So when we talk about DNA cloning, again, that's the process of making millions of copies of a specific sequence of DNA. Could be a gene, could be a promoter, could be any sequence that you are interested. Why do you do that? What is the point of doing cloning? Well, is trying to basically the ultimate goal is could be many of it. Here you have some example here. You could be cloning a sequence, make millions of copies of that to use it later on, for example, to produce a protein that is coded by that gene, right? For example, insulin is made this way. I'll show you an example later on of the human hormone, growth hormone, how is made that one. It can be used to produce vaccine. Well, the spike protein from SARS-CoV-2 as being cloned inside adenoviruses, right? Which then is used to get humans to produce the spike protein and therefore vaccinate them. You can use it to express a protein within plants or other organisms to improve them in different ways. For example, with plants, you want to make them more resistant to infections, or for example to drought. And then, or simply, you might do it just because you want to study how a promoter works. You clone it, make a copy, and see how you can turn it on and off. Does it work, or how it's expressed a gene and so on. So different purposes of doing cloning. Now, how is cloning done? Is basically you take a specific sequence. Now, this sequence can be taken out of a genome by cutting restriction enzyme, for example, or can be produced by PCR. We'll talk about that, how it works later on. And then you put it inside a different DNA molecule. So imagine this being the gene that you cut out from the genome. Then you insert it into a different molecule, which is called a vector. And then you produce this mixture of your target sequence or sequence of interest, the vector, and this is referred to as recombinant DNA. Again, then you would take this recombinant DNA, you would insert it either within the final target, or if you want to produce more and more, into a bacterium through a process referred to as transformation. Here is an example of why you would do this cloning and how you would do it. This is, for example, to produce how is produced the human growth hormone. It's a hormone that is normally produced by your pituitary gland. So You can take out the gene from actually human cells. You can take it out by cutting out with restriction enzyme, for example. This will be the insert. Or you can do that by PCR. You design primers on the side, you make millions of copies and so on, then you cut it. Then you would insert it into, in this case, is a plasmid here. This is a vector. That's what we refer as a vector. This could be opened up with the same restriction enzyme used to cut the insert out. You cut it out, and I'll show you how it works later on. And then you put them together, and you produce the recombinant DNA plasmid, recombinant DNA molecule here. Now, if this molecule here has a promoter, you need to remember those sequences that turn on and off genes, and a termination sequence here, and you have the gene here now without introns. In this case would have to be without introns, because then you put it inside a bacterium here. Now this bacterium will start actually producing this protein. So in this case, the growth, human growth hormone out of that. Again, it should be without introns, because as you know, bacteria do not do splicing. That's one example of an application. Now, so the insert can be in a gene, can be the product PCR, different things. The vectors. The vectors, there are different type of vectors. First of all, they can be considered as cloning or expression vectors. Cloning vectors are just to have just the simple goal of being used just to make millions of copies of a specific sequence that you clone inside. That's it. Whereas expression vectors have the goal to actually express what that sequence contains. You want to express the genetic content of that sequence. So if you cut out the RNA coding sequence from a transcription unit, you will put in an expression vector. You would express that gene. Okay. Now the plasmid can be of different types. Here we'll talk about only about plasmids, simple ones, commonly used in the lab, and that's what you'll use in the lab, one of your first labs. So the difference between them are multiple. Okay, it's where they can actually be transcribed, if it's an expression vectors or not. One difference that I'm focusing on here is the how big the insert can be. So plasmid can take usually smaller insert, up to 10,000 kilobases. It's fairly rare to have a plasmid that can take this, but that's how big it can be maximum. Viruses, that can be lentiviruses, adenoviruses. Like the adenovirus is the one, for example, used to produce the AstraZeneca vaccine, or the Johnson & Johnson one, the Sputnik. They're all made with viruses. Or bacteriophages are viruses that infect bacteria. Now they can take larger molecular DNA, up to 20 kilobases. And then you can even use artificial chromosome, like the yeast artificial chromosome here. This is a whole chromosome that actually, once you insert it inside even a eukaryotic cells, it will start dividing like the other chromosomes. So it will start undergoing mitosis like the other chromosomes. And the big advantage of this chromosome is that now you can insert huge sequences of DNA inside. So many types of vectors here. Here we'll focus on the plasmid for simplicity, and also because they're very commonly used as well. Now, how you choose the vector among that section there, it depends on different things. Again, as we say, the size of the insert is a big factor. Then what are you going to use that for? If you want to just express a protein in bacteria, go straight to plasmid. It's easy. Or whether you want to achieve short or long-term expression. We'll talk about gene therapy in the next lecture. And that's a big issue there. If you want to permanently correct a genetic disease, you want to have long-term expression. If instead all you want to do, let's say, is kill some cancer cells, gene therapy, you just want a short-term expression, right? That's all you need for a vaccine. You'll get a short-term expression. So what you use, so you want to use a long-term expression, then most likely you want to use like something like an engineered retrovirus. It would insert in your DNA, and then once it's there, you can get it out. It will keep producing the protein that you want. All right. But if you want to just have a transient one, you can use a plasmid, you can use other type of vector. Focusing on plasmid DNA. Now, this plasmid DNA molecule, I anticipated that earlier on, are circular molecules of DNA which are extrachromosomal. They're normally found in bacteria, but they are not part of the bacterial chromosome. They are completely independent from the bacteria. In fact, they even have their own origin of replication. So they keep replicating independently from the bacterial chromosome. And in fact, you can even start with a very few plasmid inside a bacterium and then go back if you go back a few days later, you can have thousands of them because they keep replicating on their own as almost independent organism. They give some benefit to the bacteria, and that's the reason why bacteria pick them up. One of them is what many of them still carry, which is the presence of an antibiotic resistant gene. So in general, so these are the common property of all plasmid. They all have their own origin of replication. They all have their own marker genes. Some of them are natural antibiotic resistant genes, like to ampicillin or other type of antibiotics. Then they also have an artificial multiple cloning site. This is a sequence you'll see later on of restriction site recognized by restriction enzymes, so which are then used to insert different foreign molecules. That's where the foreign molecules will be inserted right here. Now, this is a picture that shows you what I told you. So these are normally found in bacteria. Again, they replicate independently from the chromosome. They have their own independent origin of replication. They carry marker genes, particularly antibiotic resistant genes here, and then they have regions where you can insert different foreign molecule, which is the multiple cloning site, which has a lot of restriction sites. Now, focusing here, so marker genes can be an antibiotic resistance. What is the point of having that? What is the advantage when you use them for cloning? Is that this allows you to distinguish the bacteria that have picked up your plasmid from those that don't have it. So for example, imagine you have inserted here a sequence of DNA in the multiple cloning site, which expresses a gene. And now you want to make sure you put it inside bacteria through a process called transformation, and you want to make as many copies as possible, right? And so you want to really select only the bacteria that do have the plasmid inside. So once you put them in growth media, these are the cells that replicate, that make more and more plasmids. You don't want to have the ones that don't have it, like this one. How do you do it? It's by adding exactly the antibiotic that this gene gives resistance to. So if this is an ampicillin resistant gene and you add ampicillin to the growth media, now those bacteria that don't have the plasmid would die. Only those that have the plasmid will be able to be resistant, and therefore you come out with a very nicely pure population of bacteria that are ampicillin resistant and therefore all have thousands of copies of your plasmid. You get a much better yield of your plasmid out of them. Other marker gene commonly used is what is called the LAC-Z gene. This is a gene that codes for this protein called beta-galactosidase. This is a protein that can convert, it's a natural protein actually found in some bacteria, but basically what it can do in the lab, it can convert a colorless compound referred to as X-gal into a blue product. Okay, so now this gene, this marker gene, usually is put so that he has the multiple cloning site, or polylinker is the same thing, inside it. Okay, so you have this multiple cloning site inside this beta-galactosidase. This is where you will clone different molecules, right? So if you don't clone anything, now you have that the beta-galactosidase gene is still intact, and therefore will still be able to convert this colorless substrate into blue product, right? However, if you now clone something inside here, now you would disrupt the reading frame of the beta-galactosidase gene, and therefore this gene with the insert inside will not be able to produce a functional beta-galactosidase, and therefore there will be nothing able to convert this colorless substrate into a blue product. Well, this allows you to select at a certain point the bacteria that are at this point they have the insert, the plasmid with the insert inside, from those that don't have it. So imagine you've done your experiment to clone your insert, whatever you want, the gene inside here, let's say. You want to figure out, or you want to purify only the plasmid that has it. Now, you could take your mixture, mix it with your bacteria, right? Now you would have different colonies forming. So each one of these dots you see right here, these are growth in a the agar is called. It's a growth media. It would have a antibiotic added to this. So all of these bacteria would have this plasmid. So if you have add ampicillin, only the bacteria that have this plasmid here will be growing. However, not all of them would have inserted what you want. Those that don't have anything inside will still be producing the beta-galactosidase. So they will still be producing the blue product. So now you know that these bacteria here, these colonies of bacteria here in blue, don't have your insert. Whereas the ones that have it are white. They don't produce beta-galactosidase. So now if you want to select the bacteria that have not only the plasmid, but also the insert that you have added, you want to just have a pure colony of bacteria with the plasmid and the insert inside, you take one of this colony, you transfer to a new, in this case a liquid media, you grow it, you're guaranteed pretty much that from that colony you'll be extracting plasmid that have exactly what you want. Okay, that's a second type, very helpful type of selection markers added to the plasmid. So all plasmid have different markers, some that gives antibiotic resistance, some of them that can show whether there is an insert or not, the beta-gal one. They all have their own origin or replication. They can all replicate independently inside bacteria, and then they all have this multiple cloning site or polylinker. What this is a sequence of DNA that has many restriction enzymes. And the reason why you want to have this sequence inside there is that now you can use any of these restriction enzymes to open this plasmid open and insert what you want. Okay, the gene, the promoter, whatever you are interested on cloning. How do you do that? Let's go through an example here. So imagine this is the sequence of the plasmid. Obviously this will be circular. What we're focusing on the polylinker. Imagine this is the polylinker and this is one of these palindromic sites that is recognized by a restriction enzyme. And now imagine this is your foreign DNA. Okay, this is the sequence that you want to clone, right? And this sequence has on both sides exactly the same restriction site that is found in the polylinker on the plasmid, right? So now what you can do there is you can first take your plasmid and you cut it open with this restriction enzyme, which is EcoR1, right? You cut it open, you digest it, so this will cut the phosphodiester bond here, phosphodiester bond here, and would open up the plasmid with these two sticky ends. You remember there is a partial single strand DNA molecule here, which is complementary to each other. You will open this one up. Now, if you do the same thing with the sequence that contains your foreign DNA, your target sequence there, or a sequence of interest, now you'll be able to actually cut this sequence out. This sequence still recognizable by EcoR1. Now, not only you have cutted out your DNA of interest here, but now this DNA molecule on the side would have actually partial single-strand DNA molecules here. They are exactly complementary to the one that you find on the open plasmid. So if you actually mix now, you would have to get rid of this green sequence, all right? You can run it on an agar gel, for example. You then cut out this band, so you have only the foreign DNA cut with EcoR1. You mix it together with the plasmid DNA that's been opened up there. Now, the complementary sequences would actually match with each other. These actually form hydrogen bond with this and vice versa. Now, last step will be, and is now mentioned in this, is that in reality it will still be missing here a phosphodiester bond, so this will be held together only by the hydrogen bond. You will be missing phosphodiester bond here and here, as well as here and here. And this will be done by adding another enzyme, which is ligase. Remember the last enzyme that takes place in the termination part of DNA replication. You would add that enzyme will form the phosphodiester bond here and here, and as a result, now you would end up having a recombinant DNA made by your plasmid, open up at the multiple cloning site with the equal R1, with inside at this point the sequence of DNA that you wanted to clone inside. Then you would take this plasmid DNA here, you will purify, get it to grow in bacteria, transform it into bacterium, purify. Imagine if this was the beta-gal gene, at this point the bacteria with this plasmid will not be able to produce beta-gal anymore, will be white. You will be able actually to select this plasmid there. Now, so what is all about this? Well, this is all about trying to engineer organism in any case. So for example, that plasmid that you form, imagine is producing a protein like the growth hormone, human growth hormone, insulin, or any other protein, and you insert it into a bacterium, you get them to produce that protein. You are genetically engineering that organism. Again, I'll use this example over and over. The AstraZeneca virus, right, that has the gene for the spike protein, is injected on you to temporarily genetically alter you, right? You start having now this virus entering in some of your cells and producing, adding this additional spike gene temporarily. Again, it's not going to stay there forever, but then you are producing spike, your immune system recognizes it, produce the vaccine. So this is exactly what is done in general in gene therapy. We'll talk about that in the next lecture. But the point of all we discuss right here is to ultimately produce, to clone sequences, oftentimes just to express some protein, and basically genetically engineer individual to produce whatever you want. This is an example of how, for example, this is done to produce the insulin gene that you buy in a pharmacy if you're diabetic. It consists actually of two subunits, okay, and both of them are cloned into a plasmid, which then is inserted into a bacteria. Bacteria would express both sequences and then will combine them together, and then you can purify the protein there. So using genetically engineer bacteria, and all of this is done using expression vectors in this case. Usually these expression vectors are plasmids, okay, similar to the plasmid that we discussed about earlier on, like cloning plasmid. They have all the same properties, so they all have their origin of replication, they all have a multiple cloning site, they all have selective marker genes. The difference between expression and cloning vectors is that beside the multiple cloning site, you would have also a promoter, and then on the other side you would have a termination sequence. So if this is the multiple cloning site, all right, so actually here this will be the multiple cloning site here. Now you would have the promoter here. This is a T7 like promoter, specific, is active in bacteria. You have a termination sequence here, so anything that's inserted at this point, now it will be expressed through the activation of this promoter, and therefore the protein in here will be expressed. And again, this was the example that we used before of the human growth hormone. That's exactly how it will be cloned. You will be using an expression vector here with a promoter upstream, termination sequence at the end will be inserted here, the gene inside here, put inside a bacterium, and then this bacterium will be producing the protein coming from the gene. So would have been genetically altered, genetically engineered to do that. So summarizing what we talked about in this section of these part of this lecture here, we talked about DNA cloning, so which is ultimately the goal is to produce millions of copies of a specific sequence of DNA, which is first inserted into a vector, right? And then that combination, for example, the plasmid with the sequence will be inserted into a bacterium to make millions of copies of that, or also to eventually express the protein coded by the genetic material that's been inserted into that plasmid. Last thing we're gonna talk about in this lecture is this technique, which is called polymerase chain reaction. You will actually perform that in the lab as well. So what is this technique? This is a technique that's been around for several decades, made by this scientist here, Kary Mullis, and basically it's a technique that is based on the concept of DNA replication. If you remember how it works, right? DNA opens up, it gets primed, it gets copied, and so on. But differently from DNA replication, it targets a specific sequence in whatever. For example, imagine this is your genome here. By polymerase chain reaction, you can make millions of copies of your specific sequence within your genome. For example, your growth hormone gene there. You could do it there. You could do it by using primers beside it. So it's a technique that allows you to to amplify specifically a sequence of DNA within a much larger sequence of DNA. What is the purpose of PCR? What is it used for? It's used for many things. It's used for DNA cloning. As I said before, you could be amplifying a sequence there, which then you put inside a plasmid. Could be used for diagnostic. That's a modified version, which is the real-time PCR or TPCR, is used exactly to detect the presence of different DNA-based pathogens, for example, or can be used for forensic studies there. How is it done? Well, it all works in a similar way no matter what you're doing. So there are several components that you use to perform your PCR, and that's exactly what you use in the lab. So all of these is done inside a tiny little tube, okay, where you would add the DNA where the sequence that you want to amplify is contained. So for example, if you had extract your genome DNA, you would add all that purified genomic DNA. That's the one that contains your target. Together with that, you would add two primers. We'll talk about that in the next slide, what they are. But these are two sequences, single-strand sequence of DNA that's similar to the RNA primers. In DNA replication, allow for the DNA polymerase, in this case will be a bacterial Taq DNA polymerase, allow to start from that point. This is exactly what allows you to focus on. So allows you to copy only this target sequence because the primers are designed exactly to bind right beside it. Okay, that's how they're used to direct the Taq DNA polymerase, the DNA polymerase, to copy only what you really want there. Together with that, obviously you would have to add also all the oxyribonucleotides, so the ATP, the CTP, and so on. Otherwise, I'm not sure how you can make DNA without them. And then the enzyme. The enzyme is a modified, it's not modified, it's actually the DNA polymerase that was discovered in this bacterium, Thermophilus aquaticus, hence the name TAQ, Taq, DNA polymerase, which actually can live at very, very, very high temperature. And the reason why is that these techniques requires also some step at very high temperature. If you were using the normal DNA polymers, like the normal bacterial or normal, one of many eukaryotic DNA polymers, at this high temperature, it will be denatured. But this Taq DNA polymers instead is resistant to denaturation at high temperature. So it can keep working even when you go at 95 degrees. And then obviously there will be some buffers that require specific pH, salt addition necessary for this enzyme to work. So these are the components that you use there. Now, as I said before, what allows you to really target and make millions of copies of a specific sequence are the primers. So these are single-strand DNA molecule, which can vary between 15 to 30 nucleotides in length, right? Which are designed to bind, to complement, form double helix, partial double helix, to a specific sequence which is beside what you want to copy, right? So if this is, let's say, your template sequence, that's what you want to copy, right? This sequence right here. Obviously, it will be double-stranded, right? So if you want to copy this sequence right here, imagine that's in this side right here. Let's just draw on this one. This is the sequence that you want to copy. Obviously, will be further down here. Now, one of the primer will be designed so that it's going to bind to the sequence right beside it. right? And it would have to have a specific orientation, right? So you remember the DNA polymerase works from 5 prime to 3 prime. So we'll be working, we'll start adding the next nucleotide here, then the next nucleotide here, and copying in this direction. So basically, by using this primer, and the same thing would happen in the opposite strand, right? The opposite strand here, the complementary strand to this one, would have the other primer designed here. So this will be at this point 5 prime to 3 prime, the complementary strand there. And so this will be the second primer right here. It will bind here and therefore will allow to copy this strand in this direction. So all this sequence here will be copied thanks to the positioning of the DNA polymerase right beside it, mediated by these primers. So the DNA primers are the equivalent of these RNA primers using, but they target exactly the DNA polymers. They tell the DNA polymers exactly where to go to start copying, right? And that's what is key to the specific copying of the sequence that you want. So how does it work, DNA polymer? So you have this mixture in this test tube with the DNA that contains the target sequence. You have the primers, you have the nucleotides. How do you get now the DNA polymers to make billions, trillions of copies of that specific sequence? You get it by getting that mixture to go through these 3 steps several times. Usually at least 30 to 35 times you repeat these 3 steps. So the first step is referred to as denaturation. That's the very first step, high temperature, you remember? And that's the reason why you need to use a temperature resistant DNA polymers. First step, it's at high temperature DNA so these basically causes, imagine that your target sequence is right here. It causes all the DNA, even the one that doesn't contain the target sequence, to denature, to open up, right? You want to give access to tag DNA polymerase to the strand to copy, right? So first step, denaturation, very high temperature. Then the temperature is brought down for the second step. Is referred as annealing. That's called like that because that's the step where the primers, and the temperature here between 50 to 65 degrees depends on the sequence of primers that you have. By lowering the temperature a little bit here, you allow for the primer to actually find the complementary sequence, the target sequence. So these primers are designed to be complemented to the specific sequence beside the target sequence here. The longer they are, the less is the chance that they bind somewhere else. So they bind here during annealing, and then the last step of each cycle is what we call elongation. Temperature goes a bit higher to 68 to 72 degrees, and this is when the Taq DNA polymerase start working very well, starting from the primers here, will start copying the target sequence in the 5 prime to 3 prime direction, right? By doing that, it will be copying the target sequence right there. Now, this cycle here, this is the temperature in one cycle, first of all, is not done by hand. In the old days, they would do it. You would take the test tube, 95, order bath at 95, then you move it to 50, 65, then 68, 72. You will go crazy. In the old days, they wouldn't even have a denature-resistant Taq polymers. You would have to add it for every cycle. But nowadays, this is actually done by a machine called thermocyclers. That's exactly like the one you will find in the lab here. You put your test tubes here, and here you just program these steps in the machine, and you tell the machine to repeat these steps for a certain number of times. Why do you want to repeat it? Is because by repeating it, you get that exponential growth of your desired sequence. So here you can see how it's going to work. So imagine this is your starting copy of DNA. Imagine you have a single copy of that molecule, just one, all right? And then you get the machine to go. So obviously you have added the Taq DNA polymers, the primers, and so on. And now you go through 1 cycle of denaturation, annealing, and extension. These 3 cycles I told you about. So now, this copy, thanks to the primers, will be both strands will be copied into double strand. So in the end, with this 1 cycle, you would have gone from 1 copy of target sequence to 2, right? Now, if you go back in temperature to 95 degrees again, and you do another cycle of this. Now, each of these two molecules here will first denature, exactly like the original one did. Then the primers will bind to the two separate strands here too, and therefore each of these original first copy from the first cycle will be copied into two. So at a second cycle here, you would have gone from one original single copy to four of them. As you can see here, there's a pattern. If you go for another cycle there, then each one of them does the same thing, becomes two. So you can see how by having multiple cycles now, you can achieve that exponential growth of the target sequence. For each target sequence, you would have 2 to the power of n copies produced, where n stands for the number of cycles that you go. So if you go for 35 cycles, starting from this single copy, you would end up having about 34.4 trillion copies of the target sequence. To a point that if you actually run into a gel, if you had started at this point, imagine this is a sequence within a genome, you load it without doing the PCR, you wouldn't see anything. But after 35 cycles, now you start having so many copies of these that would actually start being visible in an agarose gel, and that's exactly what you'll be doing with the in your lab. Now, what are the application of PCR? That can be used for different things. So it can be used, for example, to if you want to compare different genomic DNA, that's exactly what you're going to be doing in the lab. You'll be using D1S80. We'll talk about that in a second. This could be used for a paternity test. It could be used for a crime scene and so on. Or if you want to clone a specific sequence, again, you can design primers beside a gene and copy it. Make millions of copies of that and then clone it. Or you can use it to detect the presence of a pathogen. We'll see an example at the end. So what you'll be using it for in the lab is an example of a forensic use of PCR. What are you going to be doing in the lab? You're going to be focusing on a specific locus. A locus that is very unique. First of all, by locus, what we mean in genetics is any sequence in the DNA that is conserved. Okay, so and it's always found in the same location in a chromosome, and it's conserved. This locus can be coding for a protein, but may actually not even be coding for a protein. So, but if this sequence is conserved in every living organism, part of a same species, let's say humans, always in the same location here, is referred to as a locus. Whether it codes for a protein or not is conserved, you call a locus. Always in the same location. The one you're working with is one type of what we call variable number of tandem repeats. These are loci, so sequence of DNA they are conserved, and they are unique, and they are called variable number of tandem repeats because what they consist of is the repetition of a specific sequence of DNA. Look at this sequence of DNA here. Can be repeated multiple times. You can have it here once, twice, 3 times, 4 times, 5 times. So if this is found in your DNA, it's conserved, always found in all individual in the same location, this will be referred to as a variable number of tandem repeats. Like for coding genes, in this case, wouldn't not be coding for proteins, would not be a coding sequence, but it's conserved. You have it, your parents had it, they pass it to you, you will pass it to your kids, and so on. It will be conserved. And as with any locus, it would exist in 2 copies. You would have a copy from your mother, one from your father. The difference could be in the number of these repeats. So you may have the one copy from your mother would have 25 of these repeats, the one from your father 37, right? These differences in numbers of repeats within the 2 copies of this specific variable number of tandem repeats locus is referred still as an allele, right? When it comes to coding genes, is 2 genes that give origin to different forms of the same proteins, right? In this case, the difference between these alleles for variable number of tandem repeats will not be in the protein that they produce, but in the number of repeats. Okay, and then you will pass this number of repeats. The VNTR, so the variable number of tandem repeats locus that you're going to be working with, is called D1S80. This is found and conserved in chromosome 1 at the very tip here of chromosome 1, right? So you have it, I have it, we all have it. Okay, it's conserved. My parents had it, they passed one to me. And so since you have two chromosome 1, you have two copies, two alleles, sorry, of D1S82. Now, these D1S80s, the repeat, this repeat here consists of 16 base pairs. That's what it is. And now you can have different number of these repeats in your alleles. You can have anything between 14 to 41 alleles. You can have 29 of them, some of them are more common, they are, and so on. Now, the possible combination now in terms of allele that you have here and here, so number of repeats in this allele and this, comes out to about 435 possible allele combinations. And most individuals, 4 out of 5, will be heterozygous, meaning that they would have different number of repeats in one allele versus the other. If you have the same, let's say you have 16 in both, you will be considered homozygous. Now, why did we get here? It's because you can actually determine the number of these repeats by PCR. So if you take your genome DNA, right, you extract it, for example, from your saliva, you extract the DNA from that saliva. The saliva would have cells which have all the chromosomes, including chromosome 1, right? So in that sample, you would have also your own D1S80 alleles. Now, if you do PCR with primers designed to meet right beside this, you can actually amplify these sequences, right? And the size of what you amplify will be proportional ultimately to the number of repeats that you have present in your alleles. I'll stop here and you'll discuss the rest in the lab. That's a part of the lab that your instructor will talk about. Okay, how do you go from the PCR product to the number of repeats? The last example I'm gonna go through here of the use of PCR is what is done nowadays with the pandemic, with the test for the SARS-CoV-2 virus here. Now, there are different tests that can be used to detect the SARS-CoV-2. There are some that are antibody-based, right, where you take a sample of blood from a patient, and basically what you look for is antibodies that your immune system would have produced against this virus if it had been exposed, right? If you are exposed, if you have an active infection, your immune system will be producing antibodies to the adaptive immunity against these virus, right? And so what you do is that you take your blood, and if you put it on a little tiny plate image here, where there is that protein belonging to the virus, now if you have the antibodies, the antibody will bind to that protein, and this protein now will change color. It's based on the binding there, and therefore if you have the antibodies, there will be a change in color. You will result positive for the presence of antibodies, and therefore you will be considered as if you have if you're positive for COVID, you have an active infections. The second test, that's the one I want to focus on, is what is called a molecular test. In that case, instead, you're not looking for antibodies, your own antibodies, but you're actually looking for the genome of the virus. And you do that using a modification, a very common modification of the PCR technique, which is called reverse transcription PCR or RT-PCR. What is different, and it works similarly but it's slightly different from the regular PCR, is that in this case, this is used when you start from RNA. If you recall from previous lectures, SARS-CoV-2 is the genome of SARS-CoV-2 is a single strand RNA molecule. And so you can't really do regular PCR on it. But first, your RNA has to be transcribed into complementary DNA. And this is done exactly, do you remember when we're talking about RNA viruses and how they replicate their genome? The same way, similar enzyme, reverse transcriptase. So first, and that's the RT. There is a reverse transcription first, and then the PCR is performed there starting from the double-strand genomic DNA. So this will be your sample taken from your nose, not a pleasant type of test. So up here, this, if the virus is present, you would have the single-strand RNA genome coming from SARS-CoV-2. This will be first copied into complementary DNA, and then this is tested for by PCR, basically, for the presence or not of the virus. So if these primers, they can actually be forming products that can be detected in terms of fluorescence, so on, fluorescent in this case. And so if you are negative, these primers, since there is no RNA and there is therefore no copy double-strand DNA, they would not be amplifying anything, and therefore you would not have an increase in signal when you read your sample with the machine. Whereas if you are positive, the primers now will start binding the target sequence. It's actually more than one that you're tested for, and therefore you start having a significant signal appearing after a few cycles of PCR. Now, the intensity of the infection will be also proportionate to how quickly, let's say, these curves appear. If you have a lot of genome, it would appear very quickly. If you don't have that much, this will be delayed, and so on. And so sometimes they comment very positive, not positive. That's what it's all about. So summarizing, we are at the end of this lecture. What did we talk about in this lecture? Is all these common techniques that are used to analyze DNA. So first we talked about restriction enzyme. This enzyme that cut specific sequences which are palindromic, called restriction sites. They're all palindromic. They can be read the same way in both strands. And once they bind to them, they cut the phosphodiester bonds there, producing either blunt or sticky ends. These are used for different purposes, for cloning or, for example, to do restriction mapping. We went through one example there, and we'll go through another few examples during a live meeting. Then we talked about how DNA molecules that being cut can be separated and detected. So we talk about gel electrophoresis and how DNA can be labeled using radioactive material or fluorescent dyes. Then we talk about cloning. So how do you generate recombinant DNA molecules? We talked about cloning, how it involves basically the productions of millions of copies of a target sequence for purposes of just making lots of these copies, for cloning, for their cloning, and so on, or to express a specific protein. So we talked about the vectors. We differentiate between cloning and the spectrum vectors. And then we talked about also the production of recombinant DNA. We went through an example of how you can use a restriction enzyme to open up the multiple cloning site or polylinker of a plasmid and insert a certain molecule. And then finally we talked about polymerase chain reaction. This very common technique, which is used for diagnostic, for cloning, for many other things, and how it allows you to amplify a very specific targeted sequence of DNA, where this specificity comes from the primers that you use, designed to bind specifically beside the target sequence.